So you have learned the different bioenergy production using different bio waste, and those bio waste could be either municipal sludge waste, all kind of solid waste, including the agricultural residues, kitchen waste, industrial waste, as well as the sludge. So whether it is a solid waste or a liquid waste, it has lot of potential of producing different bioenergy. Now all of you know that what are different bioenergy. It can be your different liquid biofuels, which is varying from alcohol to terpenes to carbohydrates, or it could be the biogas, biomethane, biohydrogen, bioethane, biobutanol, and so on. You have been shown lab size reactor to the pilot plant and the working reactors of 1000 liters of capacity to produce these biofuel at a rate which is industrially exploited as well as it is commercialized as well. I discussed with you the current status of different bioenergy coming out of bio waste, and then you people understood the cost economics of these waste to bioenergy conversion. As well as you people have been told about the challenges which are still there for its production and commercialization. Okay, so what I have planned today, I have planned two laboratory sessions. One laboratory session will be dedicated to the conversion of bio waste to biohydrogen. Reason being is that you will come to know how these microbial sources can be used for conversion and what would be the yield, what would be the production rate, as well as what would be the substrate conversion rate. To understand what here the substrate is. Substrate is your complex waste. Why I am saying it a complex waste? Because it may contain different biomolecules in its polymeric form, which could be your carbohydrates, proteins, or lipids, along with that, definitely the vitamins as well. So, whenever you are starting this in your laboratory, the first requirement is the which microbe you will use for the specific biofuel production. Remember those slides which I already told you that mentioned you the different prokaryotes and eukaryotic cells can be used for these studies, varying from microbes, archaea, algae, fungi. Okay, why? Because these are the factories of different enzymes. So definitely you should have a knowledge that which organism you are using for the bio waste to bioenergy conversion. So to start with, you must be equipped with the some microbiological techniques. Either you can buy from the culture collection centers the pure microbe, or you can isolate these microbes, and you can isolate these microbes from the industrial zone or the effluents from where or to which you want it to degrade. If suppose you are isolating from the oil spill region, then definitely the microbes which are growing in this oil rich effluent would be the microbes which will be very happily degrading the different fats or oils. Likewise, if you are isolating the microbes from dairy effluent, then definitely you will get those microbes which would be very happily utilizing the residual lactose and the proteins present in the whey, which contains huge amount of chemical oxygen demand, because there, in short, it is called COD. So what happens, there is a group of microbes which will very happily digest these organic sources present in the whey, and many of them could be facultative or it can be obligate. Why I am saying it is facultative? Because at the surface or near surface, in the low oxygen tension, relatively low in comparison to the surface, they can grow happily. But when you go at the bottom, then you will see that they are specifically obligate anaerobe. They are growing in minimal oxygen or nil oxygen. So the first step of laboratory culture is to isolate the pure culture. So I would now share this video. It has been prepared by my research scholars. They did a good amount of uh, work to show you how to purify these microbes from the industrial waste or effluent. It is one of the very popular method of purifying the microbes, and this is called serial dilution technique. As you see that there are different tubes. In all those tubes, the buffer has been kept, or simple phosphate buffer, or you can take saline. Saline that will help the microbe not to get any osmotic shock, or buffer that they also play the same role. So whenever you are taking the effluent and you are diluting it in the saline in the tenfold dilutions at every step. Suppose in the first tube, if you have taken 1 ml of the effluent sample and 9 ml of water, so total it is 10 ml. Mix it properly and then transfer again 1 ml to the next tube. So the difference between the first and second tube will be the tenfold, because the first tube will be regarded as 10 to the power minus 1 dilution, because from 1 ml of the culture you diluted it with the buffer or saline to 10 ml. And then from this first tube you are taking 1 ml and mixing to the another tube. So here also you are diluting it to the tenfold. So this will be considered as 10 to the power minus 1 dilution. This will be 10 to the power minus 2, and then and so on. So you will see that 2 is minus 2, third tube will be the 10 to the power minus 3 dilution, and fourth will be the 10 to the power minus 4 dilution. Then fifth tube will be having 10 to the power minus 5, then minus 6, minus 7, minus 8. So you have to dilute it up to minus 8 or minus 10 dilutions, and then use this 10 to the power minus 8 tube, and then you have to do the spreading. You take 100 microliter of the culture and spread it over a Petri plate. I suppose all of you will be aware of this Petri plate. If not, I will tell you that it is a kind of disc, and that disc is having a lid also. You can commercially buy the sterile Petri dishes, or you can use the glass dishes. If you are using glass dishes, they are washable. and then you have to sterilize it in an autoclave to make it microbe free. And here you have to put the media. The media is a nutrition for any microbe to grow upon it. So this media usually is the Luria agar. This Luria agar actually contains yeast, peptone, sodium chloride, and neutral pH, along with the solidifying agent, which is called agarose or agar, depending upon the type of media you are using. So in the market, it is present in the name of the Luria agar. You can use many more media also. It is also present. You can also use some complex media also. If suppose you are interested in isolating the microbe, say oil degrading, then you may put some oil in it. So microbe will grow and use that oil as a food. So you have to spread that diluted culture over this plate. So what will happen after spreading? You will cover it with the lid. This L shaped, it is the spreader. So she will just cover it and then put it in the incubator. Incubator provides the specific temperature, and as well as there is no interference of outside air. So there you will find after 18 hours, you will see that there are distinct colonies. That distinct colonies can be picked up, and those distinct colonies are the group of microbes you obtain. As you can see here, I'll come to that. Here you see this plate. Here she has overgrown it. It is a pure culture actually. So once you get the distinct colonies, then you have to grow that distinct colonies in the broth culture. Broth culture is the liquid culture, and when you grow it in the broth culture, it grows in overnight from 16 to 18 hours. Then again you just plate it over the new plate so that pure culture is maintained. And that pure culture you can maintain in the laboratory either in the liquid condition or in the solid condition. So whenever you have to do the experiment, you have to always prepare a log phase growing bacteria. Log phase, it means all the cells are in the good health condition and they are replicating, means they are dividing at its maximum rate. So what happens after inoculation? She is keeping it in a shaker. So in the shaker that will grow happily because it is being shaked. Then you will see that there is a happy growing microbe and with the little frothing at the top. So here one can measure its specific growth rate. I know that many of you are engineering student may not be knowing this. This growth rate actually specifies that how fast that microbe grows. So in 1 hour, what is the growth rate? It is mentioned. So now you have the pure culture, and this culture is specific for specific waste conversion. Okay. So what will happen if you know that if it is growing happily, and through the literature you came to know that this microbe is good for utilization of, let us say, kitchen waste, then naturally you will have the kitchen waste. Then you will wash, you will dry it. First you will chop it and dry it, and then you will make a powder, and then using that powder you will have to put the some buffer and some vitamins for microbe to grow happily in healthy condition. Then you will autoclave it because you wanted to assess The what is the pure culture performance. If you are not interested in it, then you may take as such, and then you can use the group of bacteria for its conversion, as I explained theoretically in my lecture. Because this lab session I have put for the optimization. Optimization is a very important word here to understand. The thing is that I will explain with a very crude example. Suppose uh, uh, any student is there, and that student, if it is given healthy food, if he or she is given healthy food and a proper room, proper conveyance, no any other socio disturbance, or in your classes or in your office, then what will happen? That grooming of that student or candidate will be optimized. That is in the perfect condition. So, what you will achieve at the end of the exam, you will see that the student has done and received a good marks. And what are the contributions for those? Those factors, they are very important. If suppose you are not giving time and proper food, so there will be many more implications. So the same happened with bacteria also. Suppose you know that if E. coli utilizes carbohydrate and convert glucose to pyruvate through the glycogenesis, then what will glycolysis? So after the glycolysis step, that pyruvate will enter and that will make either lactate or it may make formate. So for if it makes the formate, then the yield of hydrogen through the formate will be very high. So what you would prefer, that pyruvate should not enter into the lactate forming path. Then definitely you have to optimize the pH. Why? Because little acidic pH favors the formation of the formate and further conversion into hydrogen, as well as the enzyme, the hydrogenase, as well as formate, lyase, the enzyme which is responsible for hydrogen formation. They act optimum, but that means it has the best activity under acidic condition, which may be pH 5 or 5.5 or even 6. So naturally you have to optimize that pH. Optimization means what is the specific pH point at which your microbe will efficiently liberate the hydrogen. Likewise, you have to understand here the enzyme which is working for this conversion is formate hydrogenase or hydrogenase. Then you should have the idea that these enzymes are proteins, and do they require any metal as a cofactor? Oh, yes, of course, they require the metal as a cofactor, and those can be nickel, those can be molybdenum, those can be iron. So you can optimize that concentration, a specific concentration of those metals also. So what will happen? You will get the optimum liberation of hydrogen. Similarly, you can also think that if bacteria is not healthy, if all the 100% bacteria are not in a good health, then yield will also decrease. Then in that case, you will look that the biomass is the number of bacteria which is working on your bio waste. It should be high. So what could be the factors which will promote the optimum concentration of that biomass? Biomass in this case, I am talking about the biomass, the weight of your bacteria. All must be healthy bacteria. So then you have to optimize phosphate, you have to optimize magnesium ion, you have to optimize um yeast, you have to optimize many more factors. So these are the factors which is responsible for conversion process when your bacteria act on your substrate. But here the important thing is that first you have to ensure whether your substrate is in degradable form or not. I also and many of the speakers have mentioned pre-treatment of the substrate. So the optimization comes in two steps. First, you optimize the pre-treatment of the process which liberates the free carbohydrates so that bacteria can utilize those free monosaccharides for further conversion into hydrogen. And now, how these are achieved? You have two pathways. One pathway says that you optimize each parameter and one at a time. For that, you have to understand what is parameter. Parameter is your condition which you want to optimize. Say if you want to optimize temperature. So keeping everything the standard media, standard other conditions, you will vary the temperature. That means you have to set your experiment in uh, at various temperature in triplicates, and then you will find out what is the temperature at which you obtain the maximum hydrogen. After that, you have to optimize the next parameter, and let us say that parameter is the pH. So what will you do? You will keep the first parameter, which is temperature, you will keep that temperature constant, and then you will vary the pH of your media. So first parameter temperature was optimized, and then you are altering the pH of your media, and for that you have set 6 or 10 experiments. Then you found out which pH is the optimum pH for the production. So now you have two optimized parameters, one is temperature and the other is pH. Then third, if you consider that I have to optimize the substrate concentration also. So if suppose substrate is your lactose, let us say for example, then you will vary the lactose concentration step by step, and then you will find out, yes, which lactose concentration, maybe 2% or 3%, is the optimum lactose concentration. Then you will find out I have now three parameters optimized. One is temperature, other is pH, and the third is substrate concentration. Then if you want to optimize another factor which is very important is called the inoculum age. So what is inoculum here? Inoculum is the bacterial culture which you are using for your experiment. And that suppose you have found out that by keeping all earlier parameter at the optimum concentration, you vary only inoculum concentration in all the experimental setup. Then you find out how much you are getting. Suppose 18-hour old culture is giving you the best hydrogen. So now you have fixed temperature, you fix pH, you fix substrate concentration, and then you fix the inoculum age. After that, you will also optimize what is the percentage of the inoculum you have to use in your hydrogen production experiment. So for that, you will keep other four factors which you already optimized at the constant level, and then you will vary the next one. So I'm just telling you the ways. So you consume lot of time and lot of labor as well as chemicals for doing the single parameter optimizations step by step. So here one methodology has been developed, and that is called response surface methodology. And that is a tool actually, and it was introduced quite early in 50s by Box and Wilson, and it is actually being developed by the collection of mathematical and statistical technique, and that helps in approximating and optimizing your experiments based on the models. So there are three models which is being practiced. One is the box Benton design, other is the central composite design, and the third one is the full factorial design. According to that, what happens? Box Benton design gives you the opportunity to work for each parameter at three levels. And this zero level is the which you assume that it is theoretically optimum level, and plus one and minus one you decide by your interest or your choice or your literature study or your experience. Okay, what should be the difference between 0 and the plus 1 and minus 1? The only condition is that the distance between these two must be the same distance. That means if suppose optimum pH is 6 and you are keeping plus 1 at 7, then minus 1 will be 5. So it will be minus 5, 6, and then 7. If suppose you have three parameters, you are optimizing, let us say, three parameters in one experiment, then you will have to just run 15 experiments at a time. And according to the notion, it is called number of runs. Similarly, the other design, which is called central composite design, which is CCD, in this case, you have five levels, and all these five levels are at a distance of from 0 it is plus 1 and plus 2, and from to the down level it is minus 1 and minus 2. The same is true here. You have to decide the equal distance at each level, either on the plus side or the minus side. If suppose you are taking optimum pH, I'm just telling you an example. For let us say it is pH, if it is pH 6, then plus 1 will be plus 7 and plus 2 will be plus 8. Sorry, 8, pH 8. So 0 is 6, plus 1 will be 7, plus 2 will be 8. And minus 1 will be from 6 to it will be 5, and then minus 2 will be from 6 to 4. So now you understand that what is the meaning of equal distance. So it requires 5 levels in CCD design. It is requires more quality prediction for the design space, and here you have to run maximum 20. So number of experiments will be 20. Beside that, RSM gives the opportunity to run full factorial design, and in this full factorial design, the software gives you the levels by its calculation, and it gives the three levels for each, and it gives you the 25 runs. So this is how one can design. I will show you how to do it, don't worry. So in this experiment, first step actually, you have to first select the design, and then you have to put the value at minus 1, 0, and plus 1. Then you create the specific response surface, and after the creating the response surface, you get the combination of the experiments at different conditions. Then you set your experiment according to the design. Then after that you get some results which is called response. So experimental response you have to enter in the table in the chart form. It is shown in your file only when you run this software. So they software predict by themselves, and then you have to calculate your experimental. Then this software provides you the analysis option. Then you do the ANOVA and regression analysis. After the regression analysis, the software gives the opportunity to see the optimization or the optimized value based on the predicted and the theoretical value, predicted and the experimental value, sorry. So that software gives you what should be the optimum value. If it is in the significant range and having a good F value, so you have to validate that optimum value and then you find out your result. Suppose you are working on hydrogen production, then naturally the amount of hydrogen or the yield of hydrogen or the substrate conversion efficiency, all these can be monitored. This is how it is done. So this now everyone knows, need not to explain it now. Different types of microbes, how do they produce biohydrogen. I am not going to discuss all these things now. It has been discussed in detail. So some of the macronutrients it is shown here. I, it is just to show you that whether it is yeast or potassium or calcium or magnesium or sodium or these are all the important nutrients. Beside that, some of the micronutrients which are required in a microgram concentration like molybdenate and then manganese and cobalt and nickel, all these are required in the microgram quantity for culture to grow happily. So here iron also it is one of the micronutrient. So to start with, first you have to prepare your waste in the powder form as I explained you. You will chop it, dry it, and then grind it like it is a coconut shell powder. This is the walnut shell powder. This is the rice husk powder. All these can be used. So coming to the design, how to work. Look, I will go. Yes, okay. Let the video run. So you are going to design the experiment through the response surface methodology. Suppose three variables which are the parameters. You can either pronounce it variables or the parameters. First one is the agro waste. Agro waste is the powder which you want to convert. And the second one is the temperature. And the third is the pH. So these are the codes given to these variables like x1, x2 and x3. For agro waste, the optimum intermediate level considered as 32.5 gram per liter. At equal distance, you see that there is a difference of it is kept as 20 and this is kept as 12, difference of 12.5, and this will be 45 gram per liter plus 1 level. Similarly, temperature 31 is kept as optimum, low level is 25 and high level is 37. pH can be kept at intermediate 6.5 and low level will be 5.5 and 7.5. So these are the levels which are chosen. So when you open the RSM, they ask that you create the response surface design. Then you have to put the parameters low level, optimum level and the high level. So once you feed in, then you have to click OK. And after that, yes, after that, you have to create the response. Select the box Banker design over here, or you may click the, you will get such type of table. And in that table, these are the combinations will be given. And according to the combinations, it will automatically display the levels. In what this is the run number 1 to run number 15 for the DBD design. So in experiment 1 you have to keep this concentration of the agro waste, this temperature and the pH. So in 15 bottles you have to prepare the ingredients accordingly as it is mentioned. So you will add and you will keep the temperature for the experimentation here, but pH you will keep it like that only. So these are the combinations in the run one. So all the 15 cubes I was telling you now, see the combination in the number one, it is -1, and 0. These are the three parameters A, B, C. So you will see that -1 of the parameter 1, which is 20. Parameter 2 is also -1, which is 25 degree. And it is not shown for the next parameter. It is 0. And this one is the 0. 0 is the optimum pH for the parameter 3. Similarly, run 2 is having 1, -1 and 0. 1 is the higher one, 45 gram per liter, and -1 is your 25 degree, and 0 is the optimum pH. Likewise, 15 combinations of runs are there. Then after preparing all the bottles, you have to autoclave. For autoclaving, you have to properly cap it with the aluminum foil. Autoclaving is the steam autoclaving. You have to autoclave the material under hot steam at the pressure of 115 pounds and the temperature is 121 degrees centigrade. It is done for 15 minutes, so it kills all the micro. And then you have to do the all the further proceedings in your laminar airflow. This laminar airflow provides you the controlled air to pass through it. And what happens actually, just a minute, hold on. Okay, I'm back. So this is a laminar airflow which is being used for all microbiological work. When you work in the sterilized condition in the presence of the flame, and air flows from your front towards outside, which is recirculated. So it is the filtered air, nothing can enter inside. So whatever you are doing, it is called in the sterilized condition. And before you operate this, you have to put the UV bulb on, or UV tube light, which is being fixed inside these laminar airflow for 15 to 20 minutes, so that it is killing all the microbes present at the surface of the stand or bottles, your pipettes, your tips, and everything. So the students are, they have just kept the serum bottles which is of capacity 60 ml. This is the most appropriate vessel which is being used for laboratory scale biohydrogen production experiments. And you know that these are the autoclaved bottles. This one here you can see. So Miss Priya is performing. She is transferring the micronutrients after the autoclaving of these substrates. And after adding the micronutrients, she will add the 18-hour-old culture of bacteria. She is working on a strain of Bacillus. Bacillus is a very robust microorganism. It is a spore-forming microorganism. So it gives a very high yield production of hydrogen. After that, one has to put the cap, and then you have to put a aluminium cap over that. That actually makes it airtight. After that, she is sparging the nitrogen gas into this bottle to replace the air which is present in this bottle. Usually, we are keeping it 1/4. This scheme here you have to understand. Here, the fermentation is going on at a specific temperature. A tube which is coming out of from the top, it is being it is being suspended below the water level so that whatever gas is coming out, it can be collected in the inverted measuring cylinder so that you can know how much water is displaced due to the accumulation of this gas. This is the schematics of the water displacement collection scheme. Here, one more uh, collector can be passed, which is the 50% KOH solution. If you want to collect the pure hydrogen, you have to pass it through that bubbler. So the KOH will absorb the CO2. So whatever gas here is collected is the pure hydrogen then. Now this hydrogen gas can be collected in the syringe as well, and then that gas has to be analyzed through the gas chromatograph, and that is done by pressing the plunger 2-3 times. So you collect the gases from all the bottles in different syringes, and then it can be analyzed by the gas chromatograph. I'm forwarding it so that you will be able to see the. As you already know what is gas chromatograph, Soumya has explained it. Here you see that this is the chart which we generated through the RSM. So whatever result you are getting, you are putting it over here, and that is in terms of ml of hydrogen per liter. So it is being converted. So later on after that you see, okay, yes. And after that you see that the ANOVA analysis will give you some results. Yes, after that you see this table. Yes. Here you see that the ANOVA table, it is the analysis of variance that study the level of significance or the effect of each parameter on your response. You should understand here response means the observation which you are taking. That is the response. So you have to see the linear effect. Linear effect means what is the linear effect of agriculture? You will see here the p value is 0.005. That means That means only 0.005 is a good level of experiment. They are getting failed out of it. It means that the effect of concentration of agricultural residue is significant on the hydrogen production. Only 5 experiments are not in your range. Rest 995 experiments are showing that they are affected with the change of agriculture concentration, your residue concentration. Similarly, temperature effect you see it is 0.002. It means only 2 experiment out of 1000 experiments they are getting affected. Likewise, pH is also significant. So the values which are less than 0.05 they are considered significant. Similarly, if you see the interaction of the agriculture and temperature and pH, they are also significant. If you see the after the square effect, if you see the two-way interaction means one variable with the another variable interaction, you will see that temperature and agriculture residues, your substrate, they are interacting and also significantly affecting your response. Similarly, if you compare all these three, then you will see that temperature and your substrate is having the more significant effect in comparison to the residue and pH and temperature and pH. So you see that lack of fit is not significant. It means it is following the model and it is as per the model you have designed your experiment and got the result. Similarly, when you see that R square value, it shows that your results are in concordance with the predicted value or not. Usually, if it is above 90%, it is considered as they are following the model. And the analysis equation which you obtain for this regression curve is the equation which you obtain after the analysis. And according to that analysis, you get some equation, and in that equation, you can fit in your value of all the three variables for all the runs, and then you can calculate the predicted value. So, this is how you can calculate the predicted result using that equation. You can do it by yourself and then you can fill in that data. So, you will see that how this RSM is simplifying your experiment. And in this RSM, you get the option of creating this surface plot. That means need not to go into detail of the calculation. There is a graphical representation of your result, and that graphical representation gives you the interactive effect of the two variable at a time, like temperature and substrate, pH and substrate, pH and temperature. And this light section intersection here, this light intersection, this is showing the value, optimum value due to the interaction of these two or these two or these two values. So such kind of surface plot, as well as you can generate the contour plots. I told you you have can optimize and see the optimized value. What is that optimized value? Earlier you put some zero value, but that was considered by you that it could be the optimum value for temperature, for agriculture and pH both, all the three. But the model is predicting that this 33.6 gram per liter is the optimum concentration of the residue, and 33.2 is the temperature, and 6.7 is the pH. Okay, and when you go for these three optimum value and the predicted result in that case will be 1493 ml per liter of the hydrogen. So it is your job now to repeat the experiment at these optimum value which is being predicted, and then you find out what value you are getting after doing the experiment. Okay, so this is how RSM can be performed. Okay, now she is trying to explain the gas chromatograph. What is this gas chromatograph? You should understand how it is being analyzed. This gas chromatograph is from the Agilent. This is the oven compartment where your column is placed, and this is the injector port and this is the detector port. So the one end of the column enters here, and from the top here sample is injected. So your sample travels across these capillary column. This capillary column is very large in size, but it is being in the form of a coil. So it travels a long distance, all the metabolites, and then based on its retention time, it comes out from the column. And here your detector is there, which is TCD detector. That detector actually uh, detect the different gases present in it. As the method is optimized here, we keep the temperature of oven 70, and then temperature of injector at 80, and then detector temperature at 110 degrees centigrade. And then you see that initially there is no peak, but peak starts coming at 2.5 minutes. It's a huge and the pure concentration of hydrogen you are obtaining at 2.5 minutes. There is no other peak, so it shows that it is the pure hydrogen. There is no peak here, it is just hydrogen only. If she would have injected little less, then you might have get the sharp peak. Okay, actually she introduced a very good amount of hydrogen into it. So this is how it can be analyzed, and after that you see the results. In the results you see there is one peak, 100%, and what is the value, and that value can be calculated with the standard hydrogen peak, and it is quantified accordingly. And another gas chromatograph which we have, which we use for organic acid detection, and for that, we usually in the metabolite we obtain acetic acid or formic acid or butyric acid. So we obtain acetic acid at 10.3 minutes, formic acid at 12.9 minutes, and butyric acid quite late. I already told you that when organism make acetic acid, when it is acetogenic, and when it makes the formic acid, it shows that your microbe is following the FHL pathway. And if it is majorly butyric acid, then there is a major concern that your hydrogen yield may decrease because you are getting reduced fatty acids, that is butyric acid. So this peak belongs to at the, this is, you can see that it is coming at 10.3. You can see the sharp peak because she introduced less amount of sample, so you can see the peak looks very sharp and defined peaks. And then you are getting another peak at 12.9, and that peak, you know that this is running, this chromatogram is running, this is coming out of the detector and giving you the results. So you see that it is the formic acid which is coming out from the column and getting detected. So this gas chromatography is a highly precise technique for estimation of the metabolites. Okay. Okay. Let it go. I'll go little fast. Okay. I'm fast forwarding it so that you can see it because it is coming out of the machine. It is the online signal she is measuring. Now we have reached to the 14 minutes now. It is increasing. There is no more peaks. Let's see if we are getting something else. Okay. So she shows that only acetic acid and formic acid is being formed. So there the peak uh, value is calculated. How it is calculated? I'll show you. We have the standard solution of the volatile fatty acids, and that is coming at different retention time, which is having different area, and this area is calculated with respect to the different standard peak, and this is the way it is being calculated because over the while it is written how much volatile fatty acid is there. So every peak is considered as the specific, like this one is for acetic acid, this one is for formic acid, and I think this 13.6 is for butyric acid, and then furthermore they are for isobutyric and valeric acid, which we are not getting in any of our experiment. So this is how the concentration of volatile fatty acid in the sample is measured, and in your test sample. So then it is calculated. So I have shown you the results of our research, some of the research, and we compared the potential of different microbes in terms of the biohydrogen production. So you see that how the pure culture is giving you the result, and how different substrates are being utilized. We have also studied the effect of nanomaterials because bacteria utilizes iron sulfate. So we prepared the nano iron sulfate as well as nano titanium dioxide to see its impact on the biohydrogen production and compared it with the mixed culture as well as for the pure culture. And we found out that nanomaterials helps in augmenting the hydrogen production. This is also showing that how kitchen waste, cheese waste, cyanobacterial mass, molasses, potato peel, bagasse, starch can all be utilized by different microbes, and they give you the good amount of biohydrogen as well. And here I would like to tell you a very important story. That reactor which you have seen is that of big size, but you can also construct micro reactor also. This micro reactor actually utilizes the encapsulation of microbes, limited number of microbes in each microreactor. So it is enhancing the efficiency of each microbe for substrate conversion, and that is achieved by encapsulating those microbes in the reverse micelles. And those reverse micelles are being formed by the use of surfactant in the water or buffer, And that critical molar ratio of surfactant to water controls the size and shape of these reverse micelles, among which this aerosol OT and the isooctane, which is the organic solvent. This combination worked at a very nice rate as well as durability. You can see the 35-fold enhancement in the hydrogen production in comparison to the control. And these microreactors are transparent, so photofermentation can be achieved very nicely utilizing these microreactors. These are some of the results of effect of magnesium chloride concentration by the single parameter optimization, where inoculum volume, inoculum age, temperature, pH, and pre-treatment condition of the substrate of kitchen waste is kept at hold. Hold means constant, and just varying the magnesium chloride and seeing its impact. Similarly, this is the impact with the ammonium chloride. You can see over here, keeping other at hold. Look at the effect of micronutrient here. Not a significant difference from lower to higher level. That means nickel is not getting toxic, however, giving the optimum at 180. And look at the iron here. There is a difference from lower to optimum level at to the higher. Higher concentration becomes toxic to the microbes. That is true. So you see here that Priya has done use. She used the banana peel here. Na, in this campus, students eat a lot of banana. She found out that yes, banana peel. First of all, it should be utilized properly. So she grinded, she dried, and then grinded it, and then prepared the powder, and then she optimized the pre-treatment process. So for optimizing the pre-treatment process, she used different parameters. What are those parameters? So she optimized what acid concentration and alkali concentration and the time of action affects the liberation of the free monosaccharides. Okay, so again recapitulate that China is leading ahead and we are at the third position in publishing the research on biohydrogen. You can see here. And if you see the worldwide hydrogen consumption, then China is consuming the most, then Middle East, then United States, and then it is coming the Asia, Europe, and then Asia, and then other. So look at this global production capacity of biohydrogen from the crops only. Here you see that from the potato is green, corn is blue, and this is rice or sugar beet. So you can see over the map only how different countries are producing the biohydrogen utilizing different bio resources, bio waste, maybe barley or sugar beet. So it has been already discussed and told to you that whatever feedstock you have in plenty, provided you are not competing with that feedstock for the human consumption or for the animal consumption, then you go for its utilization and convert it to the energy. The suitable energy, which can be your ethanol, like Brazil has laid it, hydrogen or biogas or bioethane, and exploit that technology which has been researched and then make it commercially viable. So the humankind will be benefited, as well as environment will be safe, because we are liberating less toxic gases. So we are conserving our biodiversity in that way. This is the pilot plant which has been developed at IIT Kharagpur, which we worked together in this mission mode project. It was funded by Ministry of New and Renewable Energy. It's a 10,000 liter pilot plant. So it shows that not only in the laboratory scale batch reactor you can produce biohydrogen successfully in large scale as well. So this technology is not just confined to the laboratory. Now it has been transferred to the sugar mill plant also in Calcutta and Kharagpur, Dhampur, which is close to the Kharagpur. So you see that it has the ports coming out of it and it is connected to the machine. So there is the online measurement. That's a real-time measurement of the production rate of metabolites as well as pure hydrogen. So this is a picture of 150 liter pilot plant. Okay. So now technology is now well established. So just we have to make it. We have to just accept it. The government has to accept it so that it can be utilized for a specific purpose, not only for research and development, also for its use, because many countries have already shown that they are using it successfully on road. This news you can see that Priya has shared that in Pune, that hydrogen fuel cell operated hybrid car which was successfully run, and you can see this is the news of October 11 this year only, Priya. And this car which has been developed, which company car is this? You can see here that this was the venture of CSIR and NCL Pune and Central Electrochemical Research Institute all together worked to make it possible that yes, we can run a car in India also. So it is not something that we are getting to know that only developed countries can do. We also have the ability of doing it. This technology, everyone already knows that New Delhi, under the flagship program of government advanced chemistry cell, batteries, and national hydrogen mission to cut down the greenhouse gas emission under Paris Climate Agreement, that Indian Railways has to run the train on hydrogen fuel based technology. So that fuel-based technology will definitely need three steps. One will be the production, other will be the storage, and then third will be the dissemination. All these three have to integrate properly to make it viable. And in Austria, hydrogen fuel cell-powered train. It's a news of 2020. So it is getting tried that how hydrogen fuel cell train can be run successfully. So they did it, and this is all about the today's lab session. Now all of you are requested to let me know that do you want to do enter the data using BBD or CCD design by yourself. I have enabled your mic. If you are interested, you can do it. Priya will share the screen and she will enter the data so that you can see the live. how it is being performed. Tell me. Hello. I'm asking to you people. I'm asking to you people if you are interested to know and perform RSM by yourself. And one thing I forget to tell you that yes, there is one more design of the RSM, that is the Placket Burman. According to the Placket Burman design, you optimize the different factors and then choose which factor is significantly affecting the process. And that is not a huge number of run you have to perform. So if suppose initially you have decided 10 factors, then design it for the Placket Burman. So all the 10 factors in different combination will be advised. Then you have to set your experiment accordingly as prescribed in the run table. Then you set the experiment and then you analyze it. Then you will see that only few of them are significantly affecting. So whatever is significantly affecting, take only those parameters or variables for further optimization by CCD or BPD. Yes, participants? Okay. Okay. Then Priya, share the table. So Priya is going to share it and I will explain. Rashi, you want to ask anything? You may
Yes,
ask.
ma'am.
Yeah.
Ma'am, I wanted to know how can we decide should we go with CCD or the another one, the model that you talked about,
Yes.
Burkett?
BBD. BBD is only having the three levels and CCD is having five levels. So if you have a manpower and if you have the facility to handle
Hmm.
these many experiments,
Okay,
because number of experiments in BBD is 15 and in CCD it is 20.
20, right.
If you are
Okay.
taking three variables and if you
Hmm.
increase the number of variables, then run will also increase.
Okay. So
Okay.
if I'm taking three variable, then I should go with BBD.
BBD or CCD depending upon your feasibility.
Okay,
If you
okay.
can run 20 experiment at a time, you go for
Right.
CCD.
Okay.
It will be more
Understood.
precise, or you can do the BBD.
Mm-hmm.
Shoaib,
मैम हाँ
yes,
मैम मैम मुझे बोलिए कि वेरिएबल्स कैसे चूज करते हैं हम इम्पोर्टेंट वेरिएबल्स कैसे चूज करते हैं
that's what I'm telling you. Suppose I told you now, whenever you are doing your experiment, you have different challenges. That is how the substrate is converted, whether the microbe is growing in sufficient number, whether the microbes are healthy or not, and if microbes are healthy, then what are the specific factors which you have to look into account for hydrogen production. So all these factors are there. So now you have to choose what you want to take up, and that you have to decide in a very strategic manner. Because if you are getting good amount of biomass that you know through the specific growth rate determination, some of the work which has been already well established, take it through the literature, and if it is totally novel, then you have to optimize all the parameters step by step manner, and that you do in the
मैम
laboratory. Yes, that you
एक्चुअली
do
आई
in
एम
the lab.
वर्किंग
Yes.
I am working on a waste and I am trying to produce a polysaccharide and I have tried many trials in this experiment in the experiments. Now I am not in a conclusion of what factors to take forward for the RSM or a CC
Okay.
design.
For that, you do first the placket Burman design. For the placket Burman design, at a time you can take 6, 7, 8, or 10 or 12 factors at a time, and you decide the run. According to the run, you put your experiment and then you analyze it through ANOVA. The software will do for you. You have to just put the command. You have to just select it, put the OK, then it will run, and then it will give you the result in terms of the significant, the P value and the F value. The
Madam,
P
which
value,
design you told? Which design?
Placket Burman,
Black
Placket,
Burman. Okay, okay.
Placket Burman. You just do it. It is very
Okay,
useful for you. It will just reduce your labor manifold and your interest also, because when you do the experiment again and again, you lose the interest. So design it properly. Then you will see that out of 10 or 15 factors, what are the main key factors which is significantly affecting your process. That is your yield of
okay.
your, what are you working? You are working on some ester production, is it?
I'm working on a polysaccharide production.
Polysaccharide. So just compare in which run you are getting the best polysaccharide amount.
Okay.
After the analysis, you will find out factor 1, 2, 3 is the most significant. Factor 4, 5 is okay, okay. And then some of them may be insignificant. So whether you control its concentration, it is not required. You understand what I mean to say?
Yes, yes,
So
yes.
if you selected 3 or 5 parameters, then you run the BBD or CCD,
Yes.
and then you will
Thank you.
optimize and see that there is a huge difference in your output. And after optimizing this, you patent it and then give this technology to the industry. Give them your microbe, give them your process, and they will use it for overproduction of your polysaccharide in large scale.
Okay.
Okay, Shoaib?
Thank you, ma'am.
Okay. Okay, Priya, share the file so that they can see it. Now Priya is sharing the RSM. How it will take few minutes. Okay. This is the table you obtain when you open the Minitab. Minitab is a software. Even Systat is a software. Just hold on. Here you see that she is clicking on uh, DOE, which is design of experiment. Then she has to open the response surface methodology. Then will be clicking on create response and surface design. Okay. Then you will obtain, after clicking that, this window will pop up. Here you have to select central composite or box beacon. You select it. You have to put the how many number of how many factors or variables you are taking, and then you click that design. Here you see the, yes, these factors. She selected 3 factors, so you will obtain these levels. First you name it. She is naming it agree, then pH, and then temperature. You have to put the 2 levels. One is low and the other is high. She is putting that level 20 and 45. Like I mentioned you, the same data she is mentioning here. Then temperature, she is writing temperature, here it is 25 and here it is 31. And then she will mention the third parameter, which is the variable, which is your pH. It is 5.5 and it is. And then create response surface design. Then and then you will see that for 3 factors it is showing that it will be having the number of runs 15. So this is the table which you will obtain, where it is mentioned in run number 1. These are the combinations, and in these combinations 15 runs will be prepared. Okay.
Excuse me.
Yeah, please speak.
Ma'am, sorry to interrupt you. Can you
No.
please start it a bit again? I actually got disconnected. Sorry, but
Okay, no problem, we'll go
thank you
back.
so much, ma'am.
Priya, go back. Start from the beginning. You have to start with the design of experiment using a software Minitab. Then select the response surface. Here put the number of design. Select the design. Put the factors. क्लियर नहीं है विंडो तुम्हारी कम. Look, design of experiment. This one, design of experiment. Her computer is hanged. बंद करके फिर से स्टार्ट करो. Just hold on ना. She'll again do it. She has to exit the program and then she has to start again. She will start the mini tab here. Go slowly so that they will follow it. Go to the design of experiment. Click the response surface. Then create the statistical design. Slowly, you go slowly. And then select the design from here. Create and then box Benton and then put the number of factors. Then we go to the display. Click the, then put the name of the factors here, and then put the low level and the high level of all the three factors which we discussed, like agriculture, then your temperature, and then pH. Okay, she's filling the low level of pH, now high level of pH. Then you click the OK, click OK, then display, and then summary analysis. Then you will see that this table will come up where it is showing the number of runs here on the left panel, and these are the combinations in every run. And then here in the results, you will, whatever result you will get, you will have to enter here. She's entering. She's entering one by one, so that how to proceed ahead you will learn. She's entering the response, whatever she obtained. So she is entering vaguely. It is not the accurate one, but she is entering it. And run number 13, 14, 15. They are showing the optimum value of all the three parameters in it. So it is in triplicate. Run number 13, 14, and 15. And after that, you have to select and then go to the slowly go to the ANOVA. क्या क्लिक कर रही हो बोलो, दिख नहीं रहा, इतना छोटा है. Go to the analysis ना. Go to the analysis. Then you select the response here. And after that you will see that slowly you have to select and then do the surface plot. So you obtain the surface plot like this. And here you can change the axis if you wish by right clicking. Again what select? All the parameters. Okay. All the parameters नहीं select किया है तो मैं agree छोड़ दिया. Okay. And then go to the response optimization plot. Then click the optimization. So this is the optimization plot you get. So again and again you have to go back to the graph only, है ना? Okay, students, participants. Hello.
Yes, ma'am. Thank you so much.
Did you follow it, na?
Yes, yes, ma'am.
You can download that software,
Yes,
and all
ma'am.
the recordings will be available to you, and we'll be sharing it to the YouTube and put the for your reference also. That is not the issue. Any other query?
Excuse me, ma'am.
Yes.
Hello. Am
Yes.
I audible?
Yes. Yes.
Ma'am,
Yes.
I'm Dr. from MIT Rajasthan.
Just a
Ma'am, I
moment.
have used. Hello.
Ah. Yes. Yes.
Ma'am, I actually I'm not expert of this field, but somebody helped me in drawing the graphs by using MATLAB
Okay.
software.
Mhm.
So in that, when I sent it for the publication, somebody replied me to mark the error bar. Is it possible to mark the error bar when we are optimizing the combination of two or three parameters together?
Error bar option is there in the rest. Hmm. Actually, what is there, na? Yeah, I don't know in the MATLAB it is available or not. But here,
Mm,
whenever we give the error, we calculate in the
yeah,
Excel, and then we report
yeah,
it
yeah.
in the form of a table. But when you go for the surface plot, you cannot do it. But maybe this
Yes,
option is
that's why
available in
I'm
the MATLAB.
asking because the reviewer commented on that there is no error bar in the graphs.
That's
I
what I'm
plotted the
saying.
graph by
In the response
yeah,
surface plot, it is not available. But
yeah.
that you can show it in the form of a table, those error bar.
Okay.
Then you
And that will be
calculate it.
normal graph. Yeah.
Yeah, that you calculate and customize it and then put your values.
Okay, okay. Okay, okay. Thank you, ma'am. Thank you very much.
Ma'am, is the software available free, ma'am, or we have to pay it?
But you can download for 1 month. It is free.
Okay. Free
For
trial.
1 month,
Okay.
it is free, yes, for the trial purpose.
Okay. Thank you.
You
Does
can
all the
download.
options will be available in free version?
Yes, yes,
To
all the options, yes.
okay. Thank you.
You have to again go to the graph and then first you have to design and then you have to put the variables. Then you will get the run structure. Then you have to do your experiment. Then you enter your response, that is your result. And then you select all the run along with the result and again select the DOE and then go to the analyze and then go to the ANOVA. You will get all the options. You just try and you will get all the options. Then you represent your data in the form of surface plot, in the form of contour plot, and
Okay.
then in the last you select the optimization, you will get the optimized values.
This Black Burman can also be done in Minitab.
In the same way, yes, in the same way. First, you have to design the Plackett-Burman design, put the number of variables, and then
Okay.
you will put the levels, and then they will give you the run structure of the run. Then according to the structure of the run, you set your experiment. And then whatever experiment you are doing, it is not only for hydrogen, for any other experiment,
Any okay. Any
for
product.
any experiment. And then just it will compare the response with the combination. And then you will have to optimize it and see what is the optimization value you are getting, as well as based on the p value and the f value, you screen which are the factors which are significant, and significant means they are significantly affecting it, and then you take those parameters for further study.
Okay. Thank you.
Any other? Any questions? If no, then
Hello.
yes,
Hello.
please. हाँ
Yeah.
बोलिए.
Sir, I'm sorry. I'd like to know how will we find out the mean value of the optimized response?
This
Means,
optimization mean this is the value where the model is giving you, that you have to validate.
okay.
Then
So
you
how
will
can we
validate.
validate
Validate
that
means that, yes, that experiment you will have to do under the optimized variable condition which you have obtained through the model. You have to do the
Yeah, yeah.
experiment in that condition, and then you will get some
Okay.
result, and that
Okay,
result
okay.
you will compare with the predicted value. Whether your result is in consensus to your model or not, whether it is close to the predicted value or not. And for that, R square value gives you the overall indication. Yes, your design of experiment is done judiciously and giving you the R square value close to the 95% or 94% or 98%. Okay.
Okay. Okay, okay. Thank you, ma'am. Thank you very much.
Okay.
Very nice presentation, ma'am.
Okay. That's good.
Thank you for the
We
ideas.
may discuss it if you wish. We can discuss it again in the second lab session also.
I need your contact number, ma'am. If
हाँ हाँ बिल्कुल,
you
you all can write my number also.
yeah, yeah, yeah.
आप लोग मेरा नंबर भेज दीजिए सबको. My student will communicate my email and my phone number to all of you.
Okay,
Okay?
okay. Thank you, ma'am. So nice of you.
Okay.
Yeah, yeah. Thank you.
Okay. Most welcome. So
So,
all may take a
ma'am,
break. Yes, yes,
ma'am, I
please.
have one question.
Yes.
Ma'am, what is the means how difference between central composite design and box bag, and when we have to select means go for central composite or box bag?
Central composite is more precise because it is selecting the 5 levels of each variable. So that gives you the closer values. However, when you go for the box Benkan, it is selecting only 3 values. So if
Okay,
suppose
but in the
what
recent
happens,
means
no, I will tell you with the example, I will tell you. Just hold on. Suppose your process is affected by pH, okay, and you are saying that level 1 is 0.5, level 2 is 1. Suppose optimum value is pH 5, which is 0 value, and minus 1 is 4.5, and minus 2 is 4. So when your optimized value comes very close, what I mean to say, there is a large difference in the response by changing the variable concentration or variable parameters, whatever, at closer points. Means if there is a small change, that small change is giving you the bigger change in the response. In that case, you go for the CCD.
okay.
है ना? You understand, try to understand.
Yes, yes.
Suppose I'm telling you there is a small temperature variation. Let us say every day we are facing morning, daytime, evening time, night time. If this difference in the night time is 2 degree, everyone sustains. And next day there is a difference in the 4 degree, that is also adjustable. And the third day, consecutive day, if there is a difference of the 10 degree, then what happens next day morning? Everybody is having a choked throat. So that you have to understand. Okay, what is the sensitivity of your experiment? Where a small change may lead to the significant change in your response, which is your output. So in that case, you will go for the CCD. That gives you the liberty of the 5 levels. It expands the, hello? Do
Yes,
you
ma'am.
listen
Yes,
me?
ma'am.
It
Yes.
expands the, yes, margin or range of your parameters or the levels of the parameters to be studied. So it gives you the more precise values. I may go closer differences, closer values, closer levels, so that that will be more accurate. So accuracy is definitely more in CCD design.
Okay. Okay, ma'am. Thank you so
Yes,
much.
yes.
Okay. Thank you, Professor Pandey. And dear participants, I will be taking a short break for lunch and then we will meet in the next session. That is also a lab session and it will be conducted again by Professor Anjana Pandey. Please take a break and join at 2:30 sharp. Thank you.